Hi, everybody. Welcome to the 2019-2020 VanBUG series. And we'll start by, yes, so we'll start with some few announcements, and then we'll have the introductory talk, the student talk by Ellie, and then after that, we'll have the feature talk by Dr. William. And then after that, we have the pizza and, sorry, this keeps on going around. We'll have pizza and drinks there in the lunchroom. Okay. So first announcement is HackSeek is happening next month. Sorry. Okay. Okay, so first announcement, HackSeek's happening next month. It's a 3-day hackathon, bioinformatics hackathon on 18th, 19th, and 20th of October. Please apply. It's so much fun. It's a great opportunity to meet other people in the bioinformatics community here in Vancouver. We have 15 different teams and different projects for this year. There's free lunch, free breakfast, and even beer on Saturday. So it's a great chance. Please apply. To check more, just talk with us after or go to HackSeek websites, and it's very easy to find. Today, our student speaker is Ellie Rich, sitting right here. And he's going to be talking about clinical computational oncology with liquid biopsies. And our main speaker is Dr. William Hsiao. He is a senior scientist at the BCCDC. And we want to thank all of our sponsors that allowed for VanBUG to happen, Ecoscope, the BC Bioinformatics program that a lot of you are part of, the BCCDC Foundation, and bioinformatics.ca, the Pacific Institute for Mathematical Sciences, Genome BC, Epsilera, and BC Cancer Research Center. And for the technical sponsors, we would like to thank Westgrid, Compute Canada, and PHSA, and all the VanBUG team. If you're interested in joining us as an organizing team for VanBUG, please talk with us during over pizza and the drinks after the talks. So with that, let's start. So there you go.
Thank you. Thanks, Laura. Wow. So, hi, everybody. Laura said, I'm Ellie. So in this building, I'm sure most people have heard of the POG project for precision oncology. Down the street towards Oak at the Vancouver Prostate Center, we have a smaller scale prostate-specific precision oncology program. We call it MyPop. A significant amount of that program is now working through liquid biopsies, so tumor DNA from the blood. In addition, there are several drug trials run by my supervisors, Dr. Kim Chi and Alex Wyatt. Some patients are genomically selected for certain trials from liquid biopsies, and others we look at the patients retrospectively with liquid biopsies. So I'm here to tell you a little bit about the bioinformatics of that today. So we have a mouse here. Oh, there we go. I see. Cool. The field of liquid biopsies is diverse. It's begun, though, to condense on some terminology. There's sort of a bisection in the field between people who study CTCs and people who study ctDNA. These things are fundamentally very different. With CTCs, you would sort and isolate tumor cells and then study those. With ctDNA, which is what we study, you study the free-floating nucleic acid in the blood. So cell-free DNA in the blood, which contains circulating tumor DNA from apoptotic tumor cells, as well as normal germline DNA. Most cancer genomics requires a match normal. So from that blood tube there, we spin down the blood. The leukocytes is where the white blood cells are, that buffy coat. That's where the match normal comes from. And then all the other nucleic acids end up in that plasma layer. So we sequence both of those, and we end up with sort of a tumor versus match normal. What kind of complicates this matter in the blood is we get a sampling of multiple metastatic sites. So you can see on this side, we have all these different tumor clones. We deal all in prostate cancer. So in addition to the germline DNA, which could appear at any percentage between 0 to 100%, what we get from the blood is all these multiple clones, and just because we see one variant with an allele frequency proportional to the green truncal clone on the bottom here, doesn't mean we also won't find stuff that's proportional to that purple clone right above it. And in fact, in those small subclones is often where we find some of these very informative variants that happen due to resistance from drugs that the patients are taking. So this is really the challenge in ctDNA. Right? When we talk about genomics, we have to talk about it in the context of the software being used to evaluate it. In sequencing regular biopsies, things with high tumor content, variant calling is a pretty mature field. It's been going on for years, and most of the variant callers will call more or less the same variants. But the current tools really aren't accurate for low allele fractions. When the variants are diluted down with reads that don't harbor those variants. There's about a dozen papers with experiments quite like this one, but I thought this one was fairly elegant. They took 96 different variants, and then they just started titrating in normal DNA and sequencing it, and we get the cross-section in the Venn diagram there of exactly how these things were called. And at 20%, only 33 of the 96 variants were called with all three variant callers. These are older variant callers, but the results generally still stand. So in order to overcome these limitations, to find all these ways of limiting amounts of error and characterizing this error, I wrote a set of tools that can be used in a pipeline or just as themselves. They're built on Dask, Numba. I don't know if anybody's into Python in the audience. A lot of CIthon in here to keep these things fast. We have variant callers, copy number callers, and as you'll see, a lot of other utilities. So this pipeline resembles other variant calling pipelines, but with the exception of the aligner and anavar for some annotation at the bottom, it's 100% written by us. We've got copy number callers, B-allele frequency stuff, even all the trimming and masking and stuff had to be done custom in order to accurately, in terms of a clinical accuracy, for variant calling in ctDNA. It really had a lot of integration of previous steps. You can see a lot of steps filter into the next steps. It's really not as linear as perhaps it once started from. Oops. Oh, there it is. So again, in any noise from low quality bases is really too much error to tolerate when trying to find variants that occur in, let's say, 1% of reads. Our trimming algorithm begins at each end, trims adapters, but then requires a bin size of n bases to contain a medium Q of greater than some number in order to stop trimming. We really, you can see here towards the end of reads, the error rates really goes up. We then also do some really heavy masking of low quality bases. The real trick to calling these somatic variants is that we developed a database stored as a Apache Arrow parquet, which contains the mean and distribution of error rates for each possible base change at each genomic location. This genomic location on the side here is a very informative one in prostate cancer. It's the androgen receptor. People take a drug called enzalutamide that binds a specific place, and they get these variants where H875 becomes something else 875. And in those situations, then they become resistant to enzalutamide. You can see here that with each base change, there's very different rates of error. C to A, for instance, has more than double the amount of C to G. And it's really at this point where we can compare things not just as tumor versus normal, but tumor versus normal versus thousands of normal samples. So determining how much circulating tumor DNA is actually present is extremely important. For clinicians, it's highly correlated with disease progression as a fairly decent proxy of tumor burden, but also it's very important for deriving copy number from log ratios, especially with the androgen receptor that I was talking about before. These guys take androgen deprivation therapy, and they end up with like 50 copies of the androgen receptor. So it becomes kind of important to find out exactly how many copies they actually have. It's really a non-trivial problem. Some groups derive these allele frequencies from mutations, some from copy number, some from SNPs. Really depends on the amount of the genome actually being sequenced for this, what actually gets used. It's really a non-trivial problem. All that clonal mess that we saw before really factors into all of this. Just to show you what the distribution of these variants really looks like, each is an exome sample here. These guys have a mismatch repair deficiency, so they ended up hypermutated. But the guys on the other side, this mouse is terrible, the guys on the other side don't. You can see just the spread of SNV allele frequencies in a lot of these samples, and just how much trying to estimate tumor fraction from this sort of mess actually is. We ended up taking an approach which basically binned each of these things. So we take the max allele frequencies of these mutations and basically find out if we can treat them basically like any other tumor, all the way down to whether they are in a 0.5 to 3% area. At that point, really small changes in our pipeline would mean they were or were not called. From this, we can then derive copy number and such. So usually we use a 73 gene panel, or 72 gene, I guess, at the time I made this slide, to find out the ctDNA fraction. And here on the blue line, we have that ctDNA fraction from that small panel from exomes, which are then displayed here. You can see where the exome actually lays with that actual panel there. One of our real sources of error seems to be, I don't have a mouse here, but this smaller guy here, you can see he was misestimated because the variants that were found were on copy number amplified chromosomes, and but we didn't have enough ctDNA fraction in order to figure out if there was a copy number there, amplification there. So just to get into the utility of this sort of thing, because this really is the result of why it's interesting. Here's a patient. The slide got a little screwed up here, but on the side here, you can see, there it is. He had a radical prostatectomy. They took out his entire prostate, and we got these 5 samples. You can see them here with their mutations, and the heat map has the allele frequencies. You can see the mutations there, generally hierarchically cluster as you'd expect them to with a clonal ancestry. But this guy was taking this enzalutamide drug, and 2 years later, you can see that we then take some cell-free DNA, some liquid biopsies, and they have so many more mutations in them, including that AR H875. So he developed this mutation, gained resistance to it, And we're able to see that not in the prostatectomy, but in the liquid biopsies. And then we're able to track over time with even more mutations through the liquid biopsy. Just one more example of stuff we see really all the time. A lot of prostate cancers have these BRCA mutations, just like breast cancer, these recombination repair mutations, and they all respond to PARP inhibition. This is a really burgeoning field in cancer therapy. But what actually we get is reversion. Sorry, hold on. There it is. Reversion mutations. So these guys either have germline or somatic variants in the BRCA genes, and then they get indels which correct the original indel later down the line. And we see those in liquid biopsies, and we often, just like in this case, we'll find many different indels with our indel caller, some at various subclonal frequencies, some really become the norm. And so this is really how we're doing precision oncology with blood samples at the Vancouver Prostate Center. I just want to thank all the people in my lab and the people who pay for some of this stuff, and you guys for listening. Thank you very much.
Sorry. If we have any questions, we could take a couple.
Yeah, it is all from our in-house samples. We actually now have separate databases because we started doing a lot of sequencing here on the HiSeq, and those ones seem to have different error rate profiles as the stuff we do on MySeq. We're up to, I think, 2,000 or 3,000 samples at this point. We actually started building up a blockchain in order to share samples with Tampere University. We have a large collaboration there, and they're using the same technologies. So now we have a constantly updating database from 2 different universities. It's also nice to use blockchain just as a... Anybody else?
Yeah.
Thanks so much for your
Thank you
time.
so much, Ellie. Okay, and now for the main talk. Sorry, guys. Okay, by Dr. William Hsiao on infectious disease surveillance in the age of semantic web, opportunities and challenges. Please welcome Dr. Well.
All right, so some disclaimers first. And one is I only accept this talk to give this talk about a week ago, and I have to scramble to put slides together. So I end up making some new slides, borrowing some slides, and which I'll show in my talk. But most importantly, for the students in my class, here's your chance for your revenge, and you can definitely not take this as the gold standard for presentation, because I haven't really vetted my slides and practiced my talk. So I'm one of the VanBUG organizers, and that's why I got roped into doing this first talk, because we were scrambling over the summer due to various reasons of inviting speakers, but we are lining up speakers for the rest of the year. And also, I did give a VanBUG talk in 2012 when I first returned to Vancouver after my postdoc, and so it's nice to be back after 7-8 years to give you an update. But also right after that talk, I was ambushed to be asked to be one of the faculty advisors and coordinators for VanBUG. So if you get invited to give a talk, be careful. Right, so back in 2012, I gave a talk titled Microbial Genomics, Pangenomics, and Metagenomics in Disease and in Health. And that was mostly based on my postdoc work. And at that time, at the end of, I sort of have some future directions and wishes, so I thought it would be a good opportunity to actually review what I said and whether those still hold true or not. And basically, I said that microbial genomics is interesting and it's important, and I do believe there's a bright future, especially more and more frequently you're seeing news about outbreaks in various regions of the world, and not just for human but also for animals and so on. So, but I also want to point out that those pathogens are the minorities of microbes, and there are a lot of interesting microbes that are beneficial as well, and the field of microbiomes really help to study that. And one of the issues I highlighted was that there are different data types. We're not just talking about genomic sequence data. There are actually other types that we should be looking into, but I stopped short of having any solutions for that. And this time around, I actually have a lot to say about that, so stay tuned. So essentially, what I was wishing for was basically better bioinformatic tools, better data integration techniques, and better understanding of the world of microbes. And my talks would basically center around these three ideas, with the last one sort of glazed over given the amount of time we have. Also, back in 2012, of course, I have to acknowledge my postdoc supervisor, and I guess most of the students, most of you that were in my class, the special topic class, I already mentioned it, but for the rest of you, it's actually quite opportune that Claire is giving a talk next month here in Vancouver, so I certainly encourage you guys to attend the talk. She's obviously a very distinguished microbiologist, but she's also a great person, so it's good to hear from her what her takes on science is and what her take on being a good scientist is about. Okay, so I put my acknowledgement slides first because lots of people to thank, and as I mentioned, these work are mainly done by people in my group, and we went from 2 people to 15 people in about 5-6 years. So a lot of people in my group to thank, and more even higher in numbers, number of collaborators, probably between 50 to 100 collaborators that we have, given the type of work that we're doing. So I can't thank each of them individually, and or even possibly list all of their names, although I think we should try that one day just for an exercise. But I do want to thank all the funding agencies that funded our research in the last few years, and some of the, of course, institutional support such as UBC, SFU, and so on. So just before we start the discussion, I want to sort of just give a quick one-slide summary of what a public health infectious disease surveillance and investigation entails. Basically, you can think of there's 2 parallel lines of investigation. One is epidemiological investigation, focused on what the exposure is, where the exposure happened, and when it happened. So people, place, and time is sort of the saying in epidemiology. And you through that process, hopefully you converge on a common exposure and you confirm the investigation by epidemiological processes or data. And then in parallel, there's laboratory investigations that isolate the pathogens from the sample and typed, which I'll explain a little bit later, that particular organism to try to differentiate pathogens or specific strains that are involved in an outbreak versus sporadic strains. However, this process relies on a lot of phone calls, a lot of paper forms, which often contain free text, a lot of personal communications by email, so on and so forth, attachment of Excel files, so on. So it's really not very efficient and very tractable. And on the laboratory side, there's a lot of the tests that are unreproducible. Imagine if you're culturing an organism, if you didn't manage to save a clone of that, then you can't really reproduce that work. and a lot of the phenotypic tests are not reliable or not sensitive. So this results in siloed databases with a lot of typos in the database, a lot of inconsistencies in the database, and also the information generated from this process travel rather slowly and unreliably. So with modern travel, it does mean that the speed of spread of pathogens also increases. So our research overarching motivation is how can we improve the efficiency and the accuracy of infectious disease surveillance and investigation in a global context. So the process from generating data, could be sequence data, could be epidemiological data, the collection of the data, the management of data, the integration of data sets, the analysis, interpretation, and the generation of knowledge and taking actions based on those information are increasingly digitized, and technology improvement has allowed better capture and store of the data, but it doesn't necessarily make it easier to integrate, analyze, or interpret the data. So while we have unprecedented amount of digital data, the information cannot be processed manually, and we rely on computers to help with that process. And there's actually a recent cover from The Economist talking about the Internet of Things, which is what I would like to talk to you about today as well. In a more digitally connected world could also benefit public health. So a study, white paper published by McKinsey showed that a more interconnected world can actually provide public health and transportation opportunities that are in the high billions to $1 trillion range. And also the same goes for health and fitness business opportunities. So turning information into not just money, of course, which is not why we're doing this, but into opportunity to improve human health, relying on these technology is very important. And I don't need to talk about high throughput sequencing. I'm sure all of you know at least something about it. But what I do want to highlight is that the generation of these genomics data not only allow us to have higher resolution and higher sensitivity laboratory tests, it also allow value-added evolutionary and functional studies of the pathogens. So it's not just the short-term benefit of being able to do better investigations, it's also the long-term benefit of being able to understand the pathogens better, to understand how it developed resistance to drugs, how it improved its fitness in a changing environment, and also importantly, how it speciates. We often thought speciation is a slow process, but when it comes to microbes, you could have a pathogen emerging quite quickly by the acquisition of new set of genes through horizontal gene transfer. And it's also known that within a single species, and arguably in microbes, species is a vague concept. In a given species, often if you knock down a particular subgroup of that species, another relative species or relative strains end up taking over and fill in the niche. It's kind of, I was listening to radio the other day about why war on drugs doesn't work, and basically the explanation was if you arrest a bunch of street level gangsters, that niche get filled quite quickly by rival gangsters, because the benefit of selling drugs is still there. You're not removing the incentives, you're just removing the players, and other players come in and take over essentially. So same thing happens in combating pathogens. So we really need to think about how we're dealing with whether we're, by treating diseases with antibiotics, are we really winning the war against pathogens in the long run. And I would argue there are probably better ways to do that. Anyway, so in terms of the bioinformatics development in the group, we first addressed the issue of sort of a bread and butter issue of when you generate a lot of data, what do you do with it? So Jun from a research associate in my group developed Secudas, which is an automated sequence data transfer and QC tool. So as you can see here, there's no pointer, right? But as you can see here, for example, each of the rows shows the a specific sample that you sequenced, and you can tell quite quickly the taxonomic compositions of the sample. And if you're expecting E. coli and you see something else, and that's an indication of contamination, it also shows you the sequence qualities just through a click of a button. So really facilitate the quick analysis of sequence data in our lab. The sort of ongoing large-scale platform project that we have been working on in the last few years is the IRIDA project, which stands for Integrated Rapid Infectious Disease Analysis platform, and it's the official genomics epidemiology analysis platform for Canadian public health system. And it started as a partnership between SFU, Fiona Brinkman, and the National Microbiology Lab in Winnipeg, Gary Van Donsler, and myself as the PIs. And over the years, we have additional people joining, and we have been quite successful with attracting research fundings to sustain this partnership, but also to sustain this platform, which I'll tell you more about. The other highlight of the platform is that it's actually actively maintained by essentially permanent employees of our public health system. So in terms of software system sustainability, we have a sort of a good way of addressing that. So the platform it's designed to be a free open source platform, and it also takes as many technological and also biological standards into consideration when building this platform, resulting in a high quality platform. It's also a distributed platform because we realize in Canada it's a federated healthcare system. So to build a single centralized platform is going to result in the platform not being actively used by the federation. So we end up building essentially a distributed platform that allow each provinces to have their own instance of the platform, so they can keep track of their data, they can perform their own analyses, and so on. And the core functions of the platform, including the management of the genomics data, the strains, and the information as well, rapid processing and analysis of the set data, and display the information or ability to download the results. And also, more increasingly, we are adding the ability to manage contextual or metadata about the samples that are being generated. And that I'll also talk more about it. So the outcome is essentially we enable the public health system to better perform sequence data management and analysis, and reduce the bioinformatics expertise needed to do that. And it also improved the reproducibility of the analysis as the platform keep track of all the actions that are performed, and through standardized workflows, the results is also more consistent and reproducible. So I'm just doing some screenshots of the platform. As you can see, that the platform is it's a web page, web-based platform with web pages that you can browse through to manage your samples. Again, each row essentially is one sample, and you can click the samples to add them to a cart for performing analysis. And then you simply have to pick one of the pipelines that have been pre-configured and pre-built for you. And by, again, a single click of button, some fairly sophisticated analysis can be performed. These include assembly of the genomes, annotation of the genomes, detecting variants of the genome, and typing of the genomes. So the platform also allowed the data to be exported for external tools to be used, and significant engineering efforts essentially went into making this as easy to use as possible. And I wanted to just highlight one of the tools that my collaborator and I developed for typing organisms. So imagine, as I mentioned earlier, that if you are trying to do an outbreak investigation, the primary task that you want to perform is to differentiate outbreak cases from non-outbreak cases. And one way to do that is by assuming that the outbreak cases are associated because they have the same type of pathogens or same subtype of pathogen. Usually it's a single species, and within the species, a subtype of it that's relevant to a particular outbreak. So you want to differentiate that from the incidental or the non-outbreak strains. In genomics, of course, by sequencing the entire genome of the organism, give you a high-resolution ways of differentiate these organisms. But as bacteria mutate quite quickly, you end up usually getting a tree that most of the strains don't look exactly the same. And so a typing tool essentially converts genomics data to a categorical data and assign it to a label or a name. And the tools that does it will then essentially reflect the epidemiological connections of the cases by typing those organisms with, by assigning those organisms with the same label. So as I mentioned earlier, that area has functionality to keep track of what you do in the system. So it not only record all the versions and all the parameters you use in your analysis, it also keeps track of all the changes to the database. And this is actually done with industrial strength Java frameworks that allows detailed logging of changes to be done automatically in the system. So overall improving, again, the reliability and reproducibility of the data. And also mentioned that it is distributed in a secure web system. Imagine, when you're doing public health analysis, you want your system to be secure so people can just log into the system and find out, who has what infection and so on, right? So while we allow different organizations to have their own instance of IRIDA, each instance is tightly controlled and on who can access the system. And again, for those technophiles out there, we use industry standards of OAuth 2 for authentication and authorization of who can access the system, including not just people, but also softwares. So you can actually write scripts to connect to the system if you have the right credential and extract data. So this allows system-to-system communication. Again, the idea of streamlining the investigation process is try to remove the human factors as much as possible on the mundane tasks, but keep people busy on more in-depth and high-level analysis, right? So the system is designed to do that. So I was talking a little bit about typing analysis and mentioned that we're taking the whole genome and convert it to a label and using that information also to then cluster the pathogens into different sequence types. The fact is that this process can be unreliable, and you can end up creating clusters that are unstable, meaning that the membership of the clusters can change depending on your threshold or the cutoff that you use. So recently, Epidemiology, who's a scientist in the Public Health Agency of Canada, demonstrated that the clustering of microbe based on these MLST typing approach can be quite unpredictable, and it could create a situation where you are misclassifying organisms based on clustering approaches for investigations. So the analysis method for public health genomics is still evolving, and this results in discrepancies in results. When you're trying to perform, say, MLST analysis but use different algorithms, it could actually change the grouping as well. So, but if you want to be able to compare your results with another, let's say US want to compare the results with Canada, then the methodology differences could actually affect the comparison. So you end up comparing apple to orange. So method harmonization and QC of the method are needed, and these are some of the outstanding issues when dealing with genomics of applying genomics to infectious disease surveillance. So I mentioned that we have improved the speed of data processing and reproducibility, but then to carry out joint investigations, we have to be able to share data, both the sequence data and also the contextual information. And as I mentioned, Emma sitting in the front, these slides are adapted from a talk that she gave last week in an international conference in Croatia. Right, so what we're dealing with is that data can come from different sources, and depending on your investigation, if you're investigating human cases versus animal cases, your requirement for the contexts are quite different, so the contextual information can be quite different. And as you go through the process of sample collection to sequencing to the bioinformatics analysis and the collection of genomic, the epidemiological and clinical data, and so on, each of those steps can have its own standards, its own data standards, and its own variations. So one of the challenges of data integration is how you can harmonize the data sets across these variable various environments, and how you can then apply them to different tasks such as surveillance, recall through samples, doing investigations, or source attribution, meaning that you're trying to find the source that's the contamination source of an outbreak, and will assess the risk of certain practice in contributing to outbreaks and so on. So some of the challenges for public health data sharing, including the lack of mechanisms for data integration and sharing between partners. As I mentioned, IRIDA is a federated system, actually allowing the different instances to talk to each other through a standard programming interface. And so we have a solution for this. It's not a complete solution. At least we're still developing Irida actively, but it addressed some of the issues associated with the mechanism of sharing data. And as I was saying, the more tricky issue is what's called semantic interoperability. In other words, can one set of data that are encoded differently in one organization and another set of data encoded differently in another organization be harmonized through shared meaning of these data sets? That's what semantic means. So our idea is that we're not going to force a single standard on everyone, but we're going to improve interoperability by allowing one set of data to be mapped or converted to another set of data. And that's what I'll be talking about for pretty much the rest of the talk, and how we use that to improve, how we use the technology, which is called Ontology, to improve curation of legacy data, and also to improve security, privacy, and ownership of data sets. So it's trying to achieve a balance between securing your data, but also allow data to be shared for public health actions. So this is, again, Emma's slide that elegantly points out that when you look at the different data sets, the same concept can actually be labeled differently. So specimen source and isolation and host tissue sampled and source all refer to the source where the sample with the specimen is derived from. And because there are different terms, computers usually cannot automatically recognize that they meant the same thing or the same concept. The other flip side of the coin is that the same word source could also be used in different contexts and for different meanings. So in the first, as I mentioned, it stands for the isolation source, but in another database, source could mean the submitting lab rather than the source of the sample. So how do you disambiguate these type of ambiguities? So we have been embracing ontology for quite a while now and have been developing some resources in this area. So ontology, simply put, is just a formal way of structuring information so computers and human can both understand the information that's encoded. And ontology, it's a building block of semantic web. That's how computers on the internet understand and reason the information that's out there. So here's an example of how ontology can be encoded. So at the high level, you can have a term called avian food product, and usually ontology is hierarchical. So more finer terms basically has a parent-child relationship with the more broader terms, typically speaking, but not always. So so you have more finer terms such as poultry food product or avian egg product that are the child terms of avian food product in an ISA relationship, as I mentioned. And they go through this hierarchy. And one thing to note is that each of the terms will then have a universal ID to denote the term. So that same ID can then be attached to different labels and used as a way of sharing the same concept. Ontology also allowed relationships to be established between terms. So for example, while this hierarchy denotes is a relationship, you can have another set of relationship talking about that avian food product has a disposition of being a transmission vehicle for certain pathogens such as salmonella, and that then conceptually link a pathogen to an isolation source, for example. So if you do this rigorously, then you can teach computer how to reason through your data sets. can't think that's what I want to say about there. So what can ontology do? I mean, I paint a pretty general picture, but if you rigorously analyze what ontology can actually do, we're simply not quite there yet in terms of teaching computers, using ontology to teach computers about all human knowledges, right? So the low-hanging fruit is that, as I mentioned, you can use ontology to standardize vocabulary, so you can assign universal IDs to the same concept. So, and each of those concepts has a specific name, a label, right? And each of the labels has a specific definition, so on and so forth. So you can use it to standardize vocabularies. You can also use it to build a body of vocabularies. So, and this is what's termed ontology biosphere, and I think the pieces here. Sorry, that I gave my cell phones for the pizza delivery guy. So, okay, back to this. So the in order to structure ontologies, you need to put different ontologies in the same paradigm, and that's usually handled by the use of an upper-level ontology. So one example of that is the basic formal ontology, which I'll talk a little bit about later. Another way is that you have a community or a consortium that all agreed on a certain set of principles in curating the information, and it then ensures some sort of consistencies across different domains of knowledge. And one that's most relevant to biomedical research is the Open Biomedical Ontologies, or the OBO Foundry. And we essentially subscribe to the principles set out by OBO Foundry and developed our ontologies accordingly. And this improved the ability for our ontology to be compatible with other ontologies, but it's still not a guarantee because humans are just too creative in terms of expressing our knowledge and information. So another issue I'll cover a bit later on is how when you exchange data, usually you're not able to accurately reproduce the intrinsic representation of one database. So many of you probably are familiar with the idea of a relational database, where the relationship of the data in that database is implicitly built into the table structure. So when you convert from one set of tables to another set of tables, there's usually not necessarily a one-to-one correspondence. Yet often we are converting between databases by using, say, by dumping the data into an Excel spreadsheet, for example. So you flatten the data, and then trying to recreate that information is not always easy. So ontology provides some ability to deal with that, but again, due to the complexity, it doesn't handle that perfectly yet. So how far can you actually go with sharing your data by use ontology? Right, so as I mentioned, the low-hanging fruit is the standardized names and definitions and so on. And you can ask people to use the same ontology, so you can map your concepts to that common ontology and use that to share information. However, if you want to create, as I mentioned, you can assign relationship to these concepts. When you want to do that, the modeling process is far from being standardized. So different people create different models, and they're not always interoperable. And also, the models can represent data at different granularity. So are you going to talk about population level concepts, or are you talking about individual level concepts? Those can be quite different, right? So if your analysis is at the population level, your ontology might be dealing with, for example, percentage of people have a certain disease. But if your analysis is dealing with individual cases, you don't really care about the fact that there's only 3% of the population have this particular disease if the patient in front of you is dying from the disease, right? So the type of information you want to encapsulate when you're dealing with individuals would be quite different when you're trying to deal with population. And again, traverse that granularity is not always easy, but it's what the hierarchical structure of ontology allows us to do to a certain degree. So as I mentioned, upper level ontology brings some harmony, but do not always guarantee full interoperability. So, so in short, we are using ontology to our best ability, but it's not the silver bullet for data harmonization. So some of the approaches that can improve the situation is promoted by Barry Smith, who's the key developer of the basic formal ontology and one of the giants in the ontology world, especially the basic formal, especially the OBO foundry. So for example, the gene ontology was initially developed by biologists, but Barry essentially took the gene ontology and fit it into a proper logical framework to make it more consistent. So he highlighted some issues such as silo syndrome, that ontology built in a single community or in an isolated situation often becomes unusable when the second community joins because they have different requirements or different expectation of how the data should be modeled. And also the ontology built to fit the current data variables or format often are not doesn't have a long shelf life because the data set, for example, data format can change quite quickly when you jump from one sequencing platform to another. So he promote, instead of the use of data variables, try to model the actual underlying entities or actual underlying concepts. And often ontologies curations reinvent the wheels and create a lot of non-compatible architectures. And sometimes that's out of necessity, but often it's just out of ignorance, really, not knowing that a standard is already out there. So what he proposed is that we should build ontologies as a suite of interoperable modules structured to provide bursts for future ontology as needed. In other words, don't build something that's overfitting to the data, but trying to build it to reflect the reality or the concept. And these can be done through trying to model the entities, the things, the process, the attributes in the data domain or the knowledge domain that you're trying to model rather than the model the data itself. And there's, again, OBO is one effort of trying to, OBO Foundry is one effort of trying to not reinvent the wheels. Okay, so in terms of our own work, Damien and Emma had developed what's called the food ontology or food-on. And it's an application ontology describing the domain of food, including the products, the sources, and the process in food. There's over 27,000 concepts or classes in the ontology, and it's currently the largest food domain ontology, ontology in the domain of food. And as mentioned, we built it to be interoperable, so follow the OBO Foundry principles. And there's a large number of faucets that are built into food-on, but to increase interoperability, we actually reuse a lot of the terms or concepts from existing ontologies, as demonstrated in the outer circle there. In addition to that, we brought in a large number of existing terminologies or categories, catalogs of food terms, and map them to food-on. So you can see on the right-hand side, there's a list of left-hand side, there's a list of common food product names and so on that are mapped to food. And this increased the ability for different organizations to map their data sets through this essentially a translator of food-related terms. So this was initially conceived to allow organizations to exchange contaminated food information for foodborne disease investigation. But quickly we realized that it has much larger applications outside of just the food safety field. So about 2 years ago, we started to work with USDA to convert their nutrition database to use foodon as the source of terminology. And the paper is published last year in Science of Food. Right, so the other large-scale effort led mainly by Emma is genomic epidemiology ontology. So we, when we want to do public health investigations and starting to look at what are the terminologies we can use to provide an end-to-end knowledge framework for genomic epidemiology, we didn't find any, so we end up building our own. This, of course, is a large-scale undertaking that involves a large number of peoples who are either contributing or are interested in this effort, and include a lot of domain experts who help us in terms of understanding some of the domain knowledges that I listed there, such as the lab analytics, the sample, how the samples are often described, how the epidemiological investigation process. We haven't tackled much of the clinical data because there are actually quite a few standards out there already, so most likely we'll just be reusing some of them. But there's actually a very good ontology in OBO for diseases and also for symptoms. So we essentially can reuse some of these developments for our effort quite effectively. But there's currently a large consortium of over, I think, 90 members now in 17 countries, something like that, that are helping us to make this a robust resource for genomic epidemiology investigation. Right, so one of the first application of GenEpio is actually to put it into IRIDA, and this is work in progress to essentially use GenEpio terms on our IRIDA platform to allow standardized vocabulary to be used. So you can see a bit of a graph here that if you start typing words in there, it would jump to relevant concepts, and you can actually then pick the relevant concepts to annotate the different samples that you have. So in this case, the cases might have certain symptoms that are of interest to the investigation. Okay, so as I mentioned, there's a lot of legacy databases out there already, and none of them are using ontology. So what can we do with those data sets? So a few years back, Gurinder, who's a postdoc in my group, joined and with a background in text mining and in computer science. So he developed a tool called LexMapper that would transform free text into ontology, and then further mapping that to arbitrary third-party categories of terms. And also because a lot of the free texts have synonyms or typos and so on, so a lot of efforts put in to correct these terms in order to map them to ontology. So I have sort of two levels of representation in terms of details. We'll just focus on the left-hand side. Essentially, samples terms are input into the system, and then they're mapped to the different lexicons, different bodies of text in order to find the closest match, and then clean up any synonyms, synonyms in this case, but also any typos or any abbreviations and so on. And then these cleaned up terms then is mapped to the ontology and ontological terms and further mapped to any third-party categories if necessary. And then this is, of course, it's a fairly complicated pipelines that handles all these tasks. And I should also mention that all our tools are publicly available, so anything that I presented, you can actually go and try yourself and complain if the case may be. So, and here you can see an example of how that works. So in terms of the actual application, so you can just open up the file and then send it to our online service, and then a few seconds later, you get your terms back that's cleaned, that's mapped to ontologies, and so on. So we're currently actually working with FDA and USDA to standardize the sample isolation source from their databases, and currently achieving pretty good accuracy of 97% or so, meaning that you know, only 3% of the cases that the concepts are mapped incorrectly. So a term is mapped to the wrong concept, and the coverage, which is depending on the underlying ontology used, is about 80%. So there's obviously room for improvement. But if you can imagine that 80% of the terms can be automatically mapped, and you only have to deal with the 20%, that will save the data analyst quite a bit of time. Right. So, and this is a fairly fast process. Essentially, it's a rule-based system, so we can map the terms quite quickly. So we can process hundreds of, tens of thousands of terms in minutes. And it's generalizable too, by using additional ontologies and developing additional rules, we can, instead of just looking at isolation sources, look at other types of data. And okay, so talking about the issue of data sharing in terms of converting from one database to another, as I mentioned, we're still in early days of dealing with that, and the current standard is very much Excel spreadsheets or even tab delimited files that are sent between analysts, and then they have to then input that either manually or if they can do it programmatically into their local databases. So that wastes a lot of time, and typically these type of exchange occurs quarterly rather than in real time, because there's just no way that epidemiologists have the time to process the data in real time. So it's like, so if we want to be able to do investigations across multiple groups in real time, we need to come up with a computational way of converting and sharing data, and that's essentially what we're striving to do. So so in order to do that, we again, Damien Dooley in my group is the lead ontologist in my group, and develop a lot of these computational resources based on ontology. So gene or genomic epidemiology entity mart, it was conceived a few years ago, and to build a platform that allowed data specifications to be published. And essentially we call it entity mart because we allow the users to shop for terms, like you go on to Amazon and shop for things. You can actually shop for ontology terms and use that to build your data specification by using standard terms. And to date, we have over 60 or so different specifications that are available, and people can take these specifications and use them, or they can expand or modify these specifications. And by using common vocabulary or common ontology, the idea is that you increase the interoperability of the different data specification while allow customization to be done when needed. Okay, so given the time, I'll probably go through these quite quickly. But so there's in order to allow the fields across different databases to be mapped, we actually have to develop a model that can handle field-level data specification and interchange. This including conversion of units, including mapping different data third-party specifications to GenIPO, for example. So here is an example you can see on the right-hand side that the given form has both a name that's assigned to GenIPO, but also shows that it's matching the third-party standard term called postnet. So called outbreak. So the outbreak identifier is matched to the outbreak in postnet database, and using this mechanism, you then can map across different databases. Okay, so actually in one of the, we drive down to Oregon from time to time to go to an ontology conference, and one of the trips back, Damien and I had a conversation about how can we visualize the complex ontological relationships or the complex relationship between these terms visually. So being a programming with Damien came up with this interesting way of representing and very intuitive way of representing ontology hierarchies. So essentially, topologically from top down, you have the parent entities or parent terms, and then you can then zoom in and zoom out and look at all these child terms and so on. And there's additional functionalities that are built in to allow you to interrogate the relationships of the different terms through a visual display. And this actually moves quite fast, and you can actually go to this website listed here and play around with yourself, picking an ontology and browse through the different terms and looking at their relationships. And it's very useful when you reuse terms from different ontologies, because you can then see how a term is residing in different ontologies and how that relates the concepts in the different domain ontologies. Okay, so last few ideas of data sharing and applying it again to public health and to animal health. So collectively, sort of we call it sort of one health surveillance by looking at animal health, human health, and also environmental health at the same time, because a lot of these foodborne diseases are actually zoonotic diseases and have an environmental reservoir. So by looking at any of the individual sectors, you're missing a lot of the diversities and a lot of the contaminated sources out there. So the idea is that we want to be able to do surveillance in all possible areas where this particular pathogen can reside. But if you do that, it involves multiple jurisdictions, such as in our case, Ministry of Agriculture will be responsible for farm animal health, whereas the BCCDC will be responsible for human health and clinical cases, right? So they might have different requirements. So we are in the process of building IRIDA to allow selectively share of metadata and or analytical results to allow different parties to work together without fully share the underlying data that may breach privacy or jurisdictional issues. So for example, if a food company is involved in some sort of contamination event, that's under the jurisdiction of CFIA, and they will be responsible for the recall and so on, but public health then would not necessarily need that information to find relevant or associate human cases. All they need to know is that certain food product has been contaminated and know that certain people consume that particular food product. So, so segregation of information for privacy and for jurisdictional reasons is not unreasonable. So we're building tools to allow that, and also to allow the data ownership to be tracked. It's interesting that the intro speaker mentioned blockchain, because we thought about using that to track data ownership, but still work in progress. Okay, so to sum up, basically we're trying to create an ontology-based data sharing ecosystem by building the analysis platform, by building the ontologies, the knowledge representation framework, by building tools that use these analogy, but also by engaging the users and by engaging domain experts to help curate the information. And with the goal essentially to train computers to understand some of these concepts in order to streamline investigation. So last slide, what about the biology? I thought say, you know, I'm trained as a biologist and I do care about biologists, and I got into public health because I care about improving human health and not just about developing really interesting algorithms, which we do. So we actually do have a lot of ongoing studies involving many of the students and trainees here that look at, for example, transmission of salmonella in the food chain in BC. Same thing for Campylobacter. Between Campi and salmonella, that accounts for 70% of all the bacterial foodborne diseases. We also have one that look at norovirus, which is the most common foodborne pathogens in the world. So we are using genomics and the data that we're generating from these routine surveillance to understand the pathogen population structure and understanding its evolutionary history. Same thing goes to AMR-related pathogens. And the other large project that I'm involved in with quite a few people is trying to detect viruses from environmental samples. And we actually recently, I never thought I would end up in the, well, our project would end up in the cover of an agriculture magazine, but the Canadian Poultry actually featured the work that we did for avian influenza because it, like, for example, in 2015, a large-scale outbreak in the Fraser Valley ended up essentially decimated the entire poultry population in the Fraser Valley. Same thing happened in the States, resulting in a shortage of eggs and so on and so forth. So it has a huge consequence in terms of the agricultural field. And we also have some microbiome studies with one of the students, Ali, here, that go back to how, again, maybe there's some beneficial microbes or some not so beneficial microbes in some of these complex diseases. So that's all. Thank you. And if you're interested in any of the resources, those are the links. Thanks. Questions? Okay, go ahead.
So given how well it works, why not? Why bother?
So there are several reasons. One is that LexMapper works well when the concepts are quite simple, but some of these terms can be quite complex, or the reverse is also true. Some of these terms are missing a lot of information. So we often see, for example, let's say bovine, and you don't know if that's talking about the actual cow, or is it talking about a product that you derive from that cow, or is talking about the environment that the cow is in, and so on. And that information is actually encoded in the study. So they sort of say, okay, I'm going to use bovine to describe this particular sample type. So what we're trying to do is, even though mapping can be done quite well, it's actually mapping. So if you have bovine, yes, it will map very well to a concept of in the ontology. So it's a correct mapping. But when it comes to actually use that, say the associated sequence data, it's really not that beneficial because it's a very vague term. So the idea is actually use this to identify concepts that are too vague and improve the curation of these metadata as well. So yes, the mapping is good, but the mapping to complex concepts is not nearly as good, and often are not very accurate either. Yeah. Yeah, that's a very good point. And in at least in public health, the idea of validating or getting your workflow accredited is fairly new. I mean, for the wet lab workflows, it's not new, but for bioinformatics workflow, it's quite new. So it's actually an active area of research of exactly what reference data set we can use, what are the pertinent parameters that need to be looked at, and so on. So it's not a solved problem, but it's an important one. Okay, if there's no more question, we can go for pizza and beer, and I'm happy to answer other questions out there. Okay, thanks.